Hi class, this is your instructor, Skylar Huff. We're now in chapter 11. This is DNA, the genetic material. So as we get to this chapter, we'll be going through the way in which genes are made, and they're made of DNA. We'll get to that structure of DNA. We'll then get to DNA replication. That's the way in which DNA will copy itself. We'll alter the genetic message by way of mutations, and then we'll get to editing genes with CRISPR. And that's some pretty interesting stuff. So hopefully you all stick around for it all. And if you have any questions, please let me know. So I'll begin here in your textbook on page 190. So as it states, using DNA at the crime scene investigation. I know you have a television, or at least have seen a TV. If you haven't seen a TV, please let me know. Nonetheless, it does say that television programs marketed as entertainment, sometimes surprisingly educational, none more than the long popular CSI programs, crime scene investigation. So the letters CSI stand for crime scene investigation, as I just mentioned. So in the spirit of this sort of drama-enhanced science education, we'll begin this chapter as detectives, tagging along with some CSI investigation, and one in which DNA, the subject of this chapter, plays a central role. So I won't read all of this to you. I'll jump right into the chapter, but I recommend you read this because it does bring to life what television tries to portray each day. So here we go. Discovering the structure of DNA. So after Mendel's work in the 1800s, a variety of different experiments in the first half of the 1900s showed that genes are composed of DNA. As it became clear that DNA is stored as the hereditary information, researchers began to question how the molecule DNA could carry the complex function of inheritance. So scientists at that time did not know what DNA molecule looked like. So it was known DNA's function, however its structure still had people flabbergasted. It wasn't yet known. So let's get to the race for the discovery of the structure of DNA. So the chemical structure. So we know DNA is long. It is a chain-like molecule made up of subunits called nucleotides. As you can see in figure 11.1, each nucleotide has three parts. A central sugar called deoxyribose, to which a phosphate group and an organic nitrogen-containing base are attached. The sugar and the phosphate group are the same in every nucleotide of DNA. However, there are four different kinds of bases. Two large ones with double rings and two small ones with single rings. The large bases are called purines. The A is for adenine, the G is for guanine, and the small bases are called pyrimidines. The C for cytosine and the T for thymine. So as we get to those, the carbon atoms that make up the sugar of deoxyribose are numbered from 1' to 5', as shown in that top panel. The phosphate group binds the 5' carbon, and the organic base binds that 1' carbon. And early in the analysis of DNA, a key observation was made by Erwin Chargaff. He noted that DNA molecules always had equal amounts of purines and pyrimidines. In fact, with slight variations due to imprecision of measurement, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. This observation led to what is known as Chargaff's rule, where A equals T, adenine equals thymine, and G equals C, guanine equals cytosine. So that suggests that DNA had a regular structure. Discovering the shape of the DNA molecule. In the 1950s, the British chemist Maurice Wilkins carried out the first X-ray diffraction experiments on DNA. In his experiments, DNA fibers bombarded with X-ray beams created a pattern on photographic film that looked like ripples created by tossing a rock into a smooth lake. So if you look at figure 11.2a, you will see this. So in 1951, Wilkins concluded from his research that the DNA molecule was composed of two strands wound around one another in the shape of a coiled spring or corkscrew. a form called a helix. In January of 1953, Wilkins shared a particularly clear X-ray diffraction photograph taken in 1952 in the lab by a postdoc, Rosalind Franklin, with two researchers at Cambridge University. So those are Francis Crick and James Watson. Using Tinker Toy-like models of the bases, Watson and Crick deduced the structure of DNA. The DNA molecule is a double helix, a structure that resembles a winding staircase. The sugar of the phosphate groups form stringers of the staircase, and the bases of nucleotides form the steps. The significance of Chargaff's rule is now clear. Every bulky purine on one strand is paired with a slender pyrimidine on the other strand. Specifically, adenine pairs with thymine and guanine pairs with cytosine. Now let's get to how DNA copies itself. What holds the two strands together? The glue is provided by weak hydrogen bonds. And those hydrogen bonds, they form between the base that face each other from the two strands. That is why adenine pairs with thymine and not cytosine. The adenine base can only form hydrogen bonds with the thymine base. Similarly, guanine can form hydrogen bonds with cytosine, but not with thymine. In the Watson and Crick model of DNA, the two strands of the double helix are said to be complementary to each other. One chain of the helix can have any sequence of bases of A, T, G, and C, but this sequence completely determines that of its particular partner helix. I'll repeat, but this sequence completely determines that of its partner in the helix. So each chain in the helix is the complementary image of one another. The complementarity makes it possible for the DNA molecule to copy itself during cell division in a very direct manner. How DNA copies itself. So the copying of DNA before cell division is called DNA replication and is carried out by an enzyme called DNA polymerase. An enzyme called helicase first unwinds DNA double helix. Then the DNA polymerase reads along each side, each single strand, and if you look closely, it's that blue strand in figure 11.4 being shown to you all in just a moment. And it adds the correct complementary nucleotide. So A pairs with T and G pairs with C, and this is at each position as it moves, creating a complementary strand. So primers. However, there are some limitations of the actions of DNA polymerase. First, it can only add to an existing strand. It cannot begin a new strand. Another enzyme circumvents this difficulty by beginning a new strand of a section of nucleotides called a primer. These are the green segments I'll now show you in figure 11.4 in just a moment. So this happens at a place where the DNA molecule becomes unzipped. That is known as the replication fork. The leading strand, or lead strand, at the replication fork, the DNA polymerase very actively shuttles several hundred nucleotides up one strand, building a new strand of DNA called the leading strand, adding onto the primer in a continuous fashion. The leading strand is the upper strand in figure 11.4, and the primer is off further to the left out of the frame of the diagram. The new strand is built when the phosphate group of a nucleotide called the 5' end attached to the sugar called the 3' end of the nucleotide, and at the end of the growing strand. In the DNA double helix, the two strands of nucleotides pair in opposite orientations, one strand running 5' to 3' and the other strand running 3' to 5'. This is why DNA is not parallel. This is why DNA is stated to be referred to as being anti-parallel. One strand in the 5' to 3' direction, the other strand in the 3' to 5' direction. The lacking strand. So a new strand is always assembled in the 5' to 3' direction because new nucleotides can only be added to the 3' end of an existing strand. Because DNA polymerase can only build a strand of DNA in one direction. It must assemble the DNA strand, called the lagging strand, in segments, called Okazaki fragments. Each lagging strand segment begins with a primer. The DNA polymerase then builds it away from the replication fork until it encounters the previous section. Now to DNA ligase. Before the new DNA molecules wind back into the double helix shape, the primers must be removed, and the segments of DNA assembled in sections on the lagging strand need to be covalently linked together. The enzyme that performs this sealing action is DNA ligase. DNA ligase joins the ends of newly synthesized segments of DNA after the primers have been removed, resulting in one continuous strand of DNA. This process is summarized at the Essential Biological Process 11A, with DNA ligase in panel 3 sealing the gaps between the DNA sections of the lagging strand. So as you can see it here, we have both the lead strand shown there in red, and then that lagging strand shown there in those fragments of red going from that left or that right to left direction from the center where a helicase is, which made those replication forks. So this is showing you how replication occurs. Chromosomes are copied in sections. Eukaryotic chromosomes each contain a single very long molecule of DNA, one far too long to copy all the way from one end to the other with a single replication fork. In eukaryotic organisms, instead, the DNA is copied in several sections of about 100,000 nucleotides each with its own replication origin and fork. So now let's get to the importance of proofreading. The enormous amount of DNA that resides within cells of your body represents a long series of DNA replications, starting with the DNA of a single cell fertilized egg. Living cells have evolved many mechanisms to avoid errors during DNA replication and to preserve the DNA from damage. These mechanisms of DNA repair proofread the strands of DNA of each DNA molecule against one another for accuracy and correct any mistakes. But the proofreading is not perfect. If it were no mistakes creating, mutations would occur. I repeat, if it were, no mistakes creating mutations would occur. Hence, errors occur and mutations do occur. So every time human DNA is passed from one generation to the next, it accumulates 100 to 200 mutations. This number, the human mutation rate, is equivalent to one mutation in every 30 million base pairs, a very low rate of proofreading error. Mutation will be discussed in more detail coming up in sections 11.3 and in chapter 14, where we consider mutation as the raw material for evolution. Now on to mutation. A change in the content of a genetic message. The base sequence of one or more genes is referred to as a mutation. I repeat, a change in the content of the genetic message, i.e., that base sequence of one or more genes, is referred to as a mutation. As you learned in the previous section, DNA copies itself by forming complementary strands along single strands of DNA when they are separated. The template strand directs the formation of the new strand. However, this replication process is not foolproof. Sometimes errors are made. These are called mutations. Some mutations alter the identity of a particular nucleotide, while others remove or add nucleotides to a gene. The cells of eukaryotes contain an enormous amount of DNA, and the mechanisms that protect and proofread the DNA are not perfect. If they were, no variation would be generated. That's the key. Mutation is the means by which variation occurs. Mistakes happen, and in fact, cells do make mistakes during replication. Other mutations occur because of DNA alteration by chemicals such as those in cigarette smoke or by radiation such as the ultraviolet light that comes from the sun or in tanning beds. Please see figure 11.5. However, mutations, they are rare. I repeat, mutations are rare. In humans, sequencing the genomes of an entire family has revealed only about 100 of the 3 billion nucleotides of the genome are altered by mutation of each generation. If changes were more common, the genetic instructions encoded in DNA would soon degrade into meaningless gibberish. Limited as it may seem, however, a steady trickle of change is the very stuff of evolution. Every difference in the genetic messages that specify different organisms arose as a result of genetic change. The message that DNA carries in its genes is the instructions of how to make proteins. The sequence of nucleotides in a strand of DNA translates into the sequence of amino acids that make up a protein. If the core message in the DNA is altered through mutation, where a thymine is in red, is inserted a guanine instead of a nucleotide during DNA replication. Then the protein product can also be altered, sometimes to the point where it can no longer function properly. Because mutations can occur randomly in a cell's DNA, most mutations are detrimental, just as making a random change in a computer program usually worsens performance. The consequences of a detrimental mutation may be minor or catastrophic, depending on the function of the altered gene. The effect of a mutation depends critically on the identity of the cell in which the mutation occurs. During the embryonic development of all multicellular organisms, there comes a point when cells destined to form gametes, or germline cells, are segregated from those that will form the other cells of the body, called somatic cells. Only when a mutation occurs within germline cells is it passed to subsequent generations as part of the hereditary endowment of the gametes derived from that cell. Mutations in germline tissue are of enormous biological importance because they provide the raw material from which natural selection produces evolutionary change. Please keep this in mind. So mutation in somatic tissue change can only occur if there are new, different allele combinations available to replace the old mutation. So mutation produces new alleles, and recombination puts the alleles together in different combinations. In animals, it is the occurrence of these two processes in germline tissue that is important for evolution, because mutations in somatic cells are not passed on from one generation to the next. However, somatic mutation may have drastic effects on the individual organism in which it occurs, because it is passed on to all the cells that are descended from the original mutant cell. Thus, if a mutant lung cell divides, all cells derived from it will carry the mutation. Somatic mutations of lung cells are the principal cause of lung cancer. Altering the sequence of DNA. One category of mutational changes affects the message itself, producing alterations in the sequence of DNA nucleotides. If alterations involve only one or a few base pairs in the coding sequence, they're called point mutations. Sometimes the identity of the nucleotide, i.e. a base substitution, while other times one or a few nucleotides are added, called an insertion, or lost, called a deletion. If an insertion or deletion throws off the reading of the gene message out of register, a frame shift mutation results. So if you look at figure 11.6, it shows a base substitution mutation, and this is what results in the change of an amino acid from proline to threonine, shown here before you. This could be a minor change or catastrophic. However, suppose that this had been a deletion of a nucleotide. That cytosine, a base nucleotide, had been skipped during the replication. This would shift the register of the DNA message, and this would be such as removing the W from the sentence using this yeah, I can't read that anymore. That is the essence of a frame shift mutation. It shifts everything downstream down because of such deletion or even an insertion. Many point mutations result from the damage to the DNA caused by mutagens, usually radiation or chemicals. The latter is of particular importance because of the modern industrial societies often release many chemical mutagens into the environment. There is, for example, considerable discussion about the possible role of chemical polyvinyl chloride, or PVC, as a potential cause of breast cancer. PVC is a key component in the manufacturing of clear plastics, like those in many plastic water bottles, and to line almost all tin cans. All evolution begins with alterations in the genetic message that create new alleles or alter the organization of genes on a chromosome. Some of the changes in germline tissue produce alterations that enable an organism to leave more offspring, and those changes tend to be preserved as the genetic endowment of future generations. Other changes reduce the ability of an organism to leave offspring. Those changes tend to be lost, as the organisms carry them contribute fewer members to future generations. Evolution can be viewed as the selection of particular combinations of alleles from a pool of alternatives. The rate of evolution is ultimately limited by the rate at which these alternatives are generated. So genetic change through mutation and recombination provides the raw material for evolution. Gene editing with CRISPR. This is where it gets good. The cute fellow you see in the photo, in your text, had its genes edited by researchers, an ability to reach in and adjust the gene message of primates undreamed of until recent advances. This new gene editing technology is having a profound impact on society. Let's get to how it was discovered briefly. The story of this remarkable new advance starts quite a while back, over 30 years ago in 1987. A Japanese biologist studying the DNA sequences of bacteria observed an odd repeating pattern within the DNA sequence at one end of a bacterial gene. A sequence of several dozen DNA bases was followed by the same sequence in reverse. Then 30 seemingly random bases of spacer DNA. This three-part pattern was repeated, with different sequences of random spacer DNA again and again. RNA molecules are copied from a DNA sequence, repeated in reverse. Those are called palindromes, and they fold back on themselves. This looped RNA can then bind to proteins, the loop sequence determining to which protein it attaches. When RNA loops produced by the palindromes discovered by the Japanese researcher were examined by other researchers, it turned out these RNA loops bind to a DNA cutting enzyme. Technically, it is referred to as an endonuclease. So there are several varieties of these endonucleases, with uninspired names such as Cas9 and CPF1. Each variety cuts DNA in a slightly different way, but all bind to palindromic loops. Please keep all of this in mind. The next step in this rapid journey of discovery happened in 2005, when the genome sequence comparisons made from databases stored on the internet revealed that a 30-base spacer DNA sequences originally identified by the Japanese researcher, thought to be random, actually were not random at all. They matched DNA sequences found in the genomes of viruses that infect and kill bacteria. The researcher had, without intending to, discovered a weapon that bacteria use to fight viruses. Now let's see, how does this work? First, the spacer RNA binds invading virus DNA of the matching sequence. Then, DNA cutting enzyme attached to the loop cuts up the virus DNA. Now, quickly came the key advance that will affect all of us. I repeat, that's us, all of us. It turns out, using now standard gene engineering approaches, any 30-base sequence can be substituted for the spacer sequence. Now, why is this a key advance? Because it allows the researcher to target any, I repeat, it allows researchers class to target any gene for modification or even destruction. This tool, which I have called CRISPR, refers to this name that is, your text calls awkward. They're clustered, regularly interspersed, short palindromic repeats. So again, clustered, regularly interspersed, short palindromic repeats. Powerful and easy to use, is rapidly altering the scientific landscape. So within a few years, a wide variety of animal cells have been CRISPR modified, including human ones. So CRISPR allows an investigator to nick a particular gene. But how does this then allow the investigator to edit the gene, changing what it does? When CRISPR-associated Cas9 or CPF1 cuts the target gene sequence, this creates a gap that the cell quickly fills in. Given that, using its DNA repair machinery to insert a new sequence of nucleotides into the gap and so restoring the genome's integrity. In a diploid organism, like a human, the alternative homolog chromosome usually provides the template for the repair machinery to copy. However, if the investigator floods the cell with many copies of a DNA sequence of the investigator's choosing, the cell's DNA repair enzymes then induce to select and insert one of these copies into the CRISPR-created gap. The result, the target gene has been edited, now containing the DNA sequence provided by the investigator. In the few years since gene editing and base editing approaches have been developed, investigators have used CRISPR systems to treat many diseases and disorders. In mice, which are easily studied in the laboratory, investigators have already successfully succeeded in using CRISPR to correct single base gene mutations responsible for sickle cell disease, muscular dystrophy, and cystic fibrosis. Work on these and other disorders in humans is proceeding at a rapid pace. So in the coming years, this novel tool is certain to have a major impact on human health. Researchers have used CRISPR to target a gene needed for a successful infection of AIDS patients. As you will learn in Chapter 16, proteins called receptors on the surface of human cells are required for the virus to enter, acting like doors to which the virus is a key. In the case of HIV, the virus that causes AIDS, the entry point is a protein called CCR5 receptor. Protein spikes protruding from the HIV virus, which you can see in the photo shown on page 201, fit in the CCR5 receptor, unlocking the door. In a highly controversial experiment, a Chinese doctor used CRISPR to disable the CCR5 in human embryos whose father had HIV. Used in vitro fertilization, these CRISPR-modified embryos developed into healthy twin girls in November of 2018. The first gene-edited humans. A particularly powerful aspect of CRISPR is that it can be used to alter all copies of a particular gene. Biomedical researchers have taken advantage of CRISPR's wide reach to solve a long-standing problem facing patients requiring organ transplants. There are 3 times as many patients needing transplants than there are available human organs. Oddly enough, pig organs would work well for many transplants, except that pigs contain a host of RNA viruses embedded within their chromosomes called porcine endogenous retroviruses, or PERVSs, that may be harmful to humans. How many PERVs, those are the porcine endogenous retroviruses, in pigs' cells? Fully 62. Too many to remove using standard approaches. But using CRISPR, researchers in 2007 succeeded in eradicating all 62 in just one step. Working with pig embryos, the researchers used CRISPR to search out each copy of the PERV gene in the embryos and used Cas9 endonuclease and snipped them out. Transferred to surrogate sows, these embryos are born PERV inactivated piglets. As adults, these pigs were a suitable source for organs for human transplant. Recently, scientists for the first time have edited genes in human embryos to repair a disease-causing mutation. The mutation, called MYBPC3, causes heart muscle to thicken and is the leading cause of sudden death in young athletes. The mutation is dominant, which you will recall from Chapter 10, means that a child will need to only inherit one copy of the mutated gene to be affected. In 2017 experiment, sperm carrying the MYBPC3 mutation were allowed to fertilize human eggs in culture, and the resulting embryos were immediately treated with the CRISPR-carrying Cas9 DNA cutting machinery. Of 58 human embryos treated, 42 were successfully edited to contain 2 normal copies of the MYBPC3 gene. In each case, CRISPR filled the blank created by Cas9, using for a template a healthy copy of the MYBPC3 gene inherited from the egg donor. In the photograph, you can see some of the resulting embryos at the 8-cell stage. This is on page 202, top left corner. While these embryos were not implanted to continue development, any adult who were developed from an embryo would be free of the disease and not transmit it to children. Repairing disease-causing mutations with CRISPR need not to be limited to embryos. In Paris in 2017, doctors succeeded in curing an adult patient of sickle cell disease by extracting bone marrow from the patient, harvesting blood stem cells, and these are the stem cells that form in a particular tissue that you will encounter in chapter 13. But what I'm getting at is, and using CRISPR to edit the DNA of the cells so they would make normal hemoglobin. So what happened is, they basically corrected the sickle cell mutation. So the edited stem cells were then placed back into the patient's bone, and that led to the production of normal hemoglobin. I told you, class, CRISPR has already changed the world. Gene drives are coming next. But firstly, let's get to cancer. So in 2017, the Food and Drug Administration, otherwise you may have heard of as the FDA, approved the first ever treatment that genetically modifies a patient's own cells to fight cancer. The therapy was approved for children and young adults with an aggressive form of leukemia, a usually fatal cancer of white blood cells. In clinical trials, 83% of the children achieved remission within 3 months. 83%. The first patient to be treated was Emily Whitehead, who was 6 years old and near death. Now 12 years old, you can see Emily in your textbook on page 202 at the bottom left. She has been free of leukemia for more than 5 years. In 2018, researchers reported that using CRISPR to repair a gene mutation in dogs with Duchenne muscular dystrophy. So gene editing system restored the production of dystrophin, that is a protein. It's a protein that is crucial for healthy muscle function in dog-treated muscle cells. About 20,000 children are diagnosed every year with this fatal inherited disorder, in which the body's muscles slowly deteriorate. Because of the possibility of unwanted DNA changes, the long-term health of CRISPR-treated dogs must be assessed before clinical trials can begin. CRISPR is wide-reaching. What about other applications of CRISPR? For example, could CRISPR be used to attack and eliminate malaria? The approach has not looked promising. For a while, a mosquito gene could potentially be modified and allowed to promote resistance to malaria. The effect would be limited to lab mosquitoes. Released outdoors, CRISPR-modified mosquitoes would not take over all populations, for the simple reason that making a mosquito unable to transmit the malaria parasite does not improve the mosquito's ability to survive and reproduce. Not part of natural selection, you all. In a sexually reproducing diploid species such as mosquitoes or us, a gene has a 50% chance of being inherited by each parent. So that without improved survival, its frequency does not change from one generation to the next. Every beginning biology student learns this as the Hardy-Weinberg rule, and we'll get to that in chapter 14. But what if you stacked the deck? If the gene passed on more than half the time, it could quickly increase the frequency within the population, couldn't it? This sort of bias, called gene drive by a professional biologist, doesn't happen often in nature, but might it be possible to find a way to do it? Most definitely. So, years ago, British geneticist Austin Burt suggested a thought experiment. Use target-specific cutting DNA enzymes, endonucleases, to attack mosquito genes necessary for transmitting the malarial parasite. Because the DNA cutting enzyme would act on both chromosomes, the effect would be to change the 50% chance of being inherited to 100%. And in fact, Burt said the target nature of the nuclease attack would drive the gene change through the population. In 2015, Burt's theoretical idea of an endonuclease drive was put to the test in Panama and succeeding in reducing wild populations of dengue fever carrying Aedes aegypti mosquitoes 93%. Gene drives really work, just as Burt said they would. Enter CRISPR, which allows for a particular gene sequence to be replaced by a new one devised in the laboratory. What if that new sequence was a two-part cassette that could not only the gene be replaced, but also a copy of the CRISPR sequence? Do you see? A gamete with this cassette in DNA will act within the fertilized egg on the DNA of the gamete contributed by the parent cell. So the CRISPR did its thing on the other gamete. Both of an offspring's chromosomes sets now contain the cassette, and all of that individual's offspring would bear that new gene plus the CRISPR cassette. In the future, every individual mated by any of these offspring will suffer the same fate, as will all of their offspring. It is a chain reaction, you all. Now imagine, again, Burt's proposal of a gene drive for mosquitoes. DNA researchers in the laboratory could insert into a mosquito a DNA cassette containing both CRISPR and a gene preventing transmission of malaria parasite, directing CRISPR to cut the original, but not edit the version of the gene. Released outdoors, this mosquito would mate with a wild individual. Their offspring inherited the wild and one from the laboratory copy of the parasite transmission gene. 50% transmission, right? But now CRISPR attacks the normal wild copy, inserting it into the place of the edited version, plus CRISPR. And so it begins, a CRISPR-driven chain reaction. When tested in fruit flies in early 2015, this sort of CRISPR-driven reaction proved to be more than just an interesting possibility. It really works. In fact, it's shockingly efficient gene drive. Using a CRISPR cassette, researchers at the University of California, San Diego, drove a recessive mutation that blocks pigmentation in fruit flies from a 50% chance of being inherited to a 97% chance. And again, a 97% chance. Geneticists are also busy at work building and testing a Burt laboratory mosquito with a CRISPR cassette containing the genes necessary to block parasite transmission. Their first job is to defeat the chromosomal repair program in the mosquito DNA that acts to counter-edit CRISPR. Then, using the CRISPR gene drive, geneticists hope to be able to drive the parasites resistant to genes into and through the wild mosquito populations, and so eliminate malaria. Field tests are scheduled to begin in Africa as soon as 2024. Over 500,000 people die every year from malaria. So what these researchers are trying to do is no small thing. And think of the range of other diseases that could be conquered using a similar approach. It is a little scary though. CRISPR gene drives would allow any scientist to spread nearly any gene alteration through nearly any sexual reproducing population. Humans don't reproduce as rapidly as mosquitoes, of course, so it would take hundreds of years for a CRISPR-driven change to spread through the human population. And yet we already hear researchers using CRISPR to modify genomes of non-viable human embryos. Surely some sort of safeguards are in order. In 2017, the United States National Academy of Science and National Academy of Medicine convened researchers and other experts to explore the scientific, ethical, and policy issues associated with gene editing research. Their conclusion? While the potential for future good is great, decisions about when and how to use CRISPR gene drives should be made collectively. If we're going to fool with Mother Nature, in other words, we should do it carefully. In October of 2020, Dr. Manuel Charpentier and Dr. Jennifer A. Dunda were awarded the Nobel Prize in Chemistry for pioneering work on the CRISPR-Cas9 system. Their work in the early development of characterizing this gene editing tool system made them pioneers in the onslaught of applications in the medical, agricultural, and biotechnological fields. So this has been your instructor, Skylar Huff. I recommend you take a closer look at this in your textbook on page 205, and also put this chapter to work as it continues on page 206. If ever you have any questions, please let me know. Thank you all for listening, and prepare well for your test.